Okay, so this is it. The PAQUi, I think that's how you pronounce it, one chip challenge. So I'm going to open this thing up, and the challenge is to be able to last at least 5 minutes after eating this one chip that has Carolina Reaper and scorpion peppers, making it hot. So again, there's the front of the box. When you open the box, it tells about their company, and you can probably pause it and check that out if you want to. The chip is in this bag. I'm not sure what it was made in 2021. That's what that's for. And then there's this brochure that introduces their other chips, and this one chip shipped along with the other hottest chips that they have, which are the haunted ghost pepper chips, which I already ate, and they are very hot, but definitely survivable. So we'll see how survivable this chip is. And so to make the 5 minutes, I'm actually going to play this video that is a video about what we're going to be learning about on Monday, this coming Monday. And by the way, this is New Year's Day, or actually this is New Year's Eve day. But on Monday, we have our first day back after break, and we're going to be learning about DNA and RNA and protein synthesis. So this video is an introductory video to DNA and RNA. And I'm going to eat this chip, start the video, and then try to hold out until the video is over. I also have something to drink here when I make the 5 minutes, when the video is over, and a bag of Cheetos, because I found that after eating the ghost pepper chips, the Cheetos really helped get rid of the heat. Okay, so here we go. Chip first. This is it. I'm nervous because they actually warn you, not to have any underlying conditions when you do this, because you could die. There it is. Oh my gosh. Whoo-wee! Smells bad. I'm going to eat the whole thing, swallow it as fast as, chew it up, swallow it as fast as I can. I'm almost on an empty stomach. I had some peanuts this morning, and it's a little after noon.
And then decided not.
All right, so here we go. It's very hot.
Sometimes it feels like DNA gets all the credit. Yes, the structure of DNA is amazing. It has a beautiful double helix structure, a twisted ladder. It has its own emoji. DNA stores genetic information and codes for your traits. However, sometimes what gets left out is how important RNA is. Without RNA, you actually couldn't get that genetic message out to your cells so that they could start producing proteins. We talk about this in protein synthesis. RNA is a very important biomolecule, just as important as DNA. In fact, RNA is even hypothesized as coming first, before DNA, in the RNA world hypothesis, something we need to make a separate video about. So let's compare and contrast DNA with RNA. First, you will find DNA and RNA in all living organisms.
Oh, so far so
in
good.
eukaryotic cells, DNA tends to be found in the nucleus, while you can find RNA both inside and outside of the nucleus.
I think he's going to be taking
Prokaryotic
his
cells
fleece off
don't
soon.
have a nucleus. Both DNA and RNA are nucleic acids,
little burp.
which are a type of biomolecule. Nucleic acids have a monomer, which, if you remember from our biomolecules video, a monomer is a building block. The monomer for nucleic acids is a nucleotide, so both DNA and RNA have nucleotides. The nucleotides of both RNA and DNA have 3 parts: a phosphate, sugar,
What's happening?
and a base. Let's draw 6 nucleotides of DNA and 6 nucleotides of RNA. DNA is generally double-stranded, and if focusing on these 2 strands of nucleotides here, you can see they run anti-parallel to each other. RNA is generally single-stranded, so you're just seeing one strand here. The sugar in DNA is deoxyribose, and the sugar in RNA is ribose. This makes sense because DNA stands for deoxyribonucleic acid. That's helpful to know because the deoxyribose is a sugar, and nucleic acid is that type of biomolecule it is. RNA stands for ribonucleic acid, as its sugar is ribose. The bases in DNA are adenine, thymine, guanine, and cytosine. It helps to remember the popular mnemonic, apples in the tree, that helps you remember that A goes with T, and car in the garage, so C goes with G, to understand how DNA bases pair. The bases in RNA are adenine, uracil, guanine, and cytosine. Notice the different one? It's uracil. So you have to change that popular mnemonic device here. Instead of apples in the tree, maybe the apples are under. Car in the garage still works though. We mentioned earlier that DNA codes for your traits, but it couldn't do that without RNA's help. In our protein synthesis video, we talk about 3 different types of RNA, and their very important roles. mRNA, which stands for messenger RNA, mRNA's job is to carry a message based off of the DNA. In eukaryotic cells, DNA generally stays in the nucleus, but mRNA has the ability to leave the nucleus to take the message to a ribosome. Ribosomes make protein, and RNA is actually a major component of ribosomes. This type of RNA is called rRNA, which stands for ribosomal RNA. Finally, we discussed transfer RNA, or abbreviated tRNA. Its job is to transfer amino acids to match the correct mRNA codon. Codon charts using mRNA codons have been developed so that you can actually see which amino acid is brought for each mRNA codon. When those amino acids are joined together, they make a polypeptide chain. Proteins are made of one or more of these polypeptide chains, and proteins have tons of different roles, but we don't want to spoil it. Check out more in our protein synthesis video. Before we go, let's try a little 3-question quiz, shall we? Just pause it after the question so that you have time to think about it. Question 1. If I have 8 DNA nucleotides, how many DNA bases do I have? And also, how many base pairs? The answer.
I'm going to pause it so you can think, but I'm also going to ask my wife to get a paper towel to help me out here. Really hot.
Each nucleotide, regardless of whether it's a DNA or RNA nucleotide, has a phosphate, sugar, and a base. So 8 DNA nucleotides would have 8 bases. DNA bases pair like this, and that's 4 DNA base pairs. Question 2. If one strand of DNA has these bases shown here, ATT, GAC, can you complete what the complementary DNA bases would be for the other DNA strand? The answer. So remember those base pairing rules for DNA and the popular mnemonic. If placing the bases in this image, reading in the direction shown on this image, they'd be TAA, CTG. Question 3. In a process known as transcription, a complementary RNA strand called messenger RNA has to complement the DNA. More about that in our protein synthesis video. So if I still had the original portion of DNA here, what would be the complementary RNA bases? The answer. So remember, it's asking for RNA. No thymine here, it's uracil instead. Apples under helps you remember A with U. Car in the garage helps you remember C with G. So in this portion, reading in the direction shown on this image, we'd have these RNA bases. Notice the answer is similar to the last answer, but there are uracil bases here instead of thymine. One last thing. Our models here, they're unable to show the beautiful 3D shape, the exact numbers of bases per turn, the chirality, but we've left you some links to explore because the goal of our videos is always to introduce the topic so that you can discover all the fascinating details and exceptions. Check out our further reading description to keep that curiosity going. Well, that's it for the Amoeba Sisters.
And that's it for me. Time for a drink. I made it at least to 5 minutes, and it has gotten better.